Hi everyone, and welcome to today's lecture. We're going to cover two very different chapters today. The first one being my personal favorite in this whole course, which is viruses and cancer. As some of you who have taken my classes before know, I love teaching microbiology, and if you like this part of the cell biochem, then make sure to sign up for my virology class this coming spring 2022. The second chapter that we will cover is then glycolysis, and we'll start going through a lot of those different cycles in the upcoming lectures. So first up, we're going to talk about viruses. Now, the first question that I pose here is, what exactly is a virus? What is it made of? And it's made of just two parts. A virus, when you hear that term, I want you to think it is just a protein capsid, which is the protein shell around it, and a nucleic acid on the inside. And that nucleic acid can either be RNA or DNA. It can be single-stranded or double-stranded. But each of the viruses, think of a nucleic acid surrounded by a protein shell. Now, the other thing you hear people talking about when it comes to viruses is questioning, are they living? And in actuality, they are not biologically considered living because they cannot do the central dogma of biology on their own without a host. And as you already know, the central dogma is DNA to RNA to protein. So DNA transcribed to RNA, RNA translated to protein. Now, as we just said, a virus is just a protein shell and a nucleic acid, which means it doesn't have ribosomes. It has to rely on a host to make viral proteins. It has to rely on a host for energy and precursors and enzymes. Okay, and so it cannot replicate and it cannot do the things of a living cell, the central dogma, without a host. Now, when you think of viruses, it's important to think about how exactly they're infecting a host cell. What's going on. The main stages that I want you to be aware of are highlighted in both of these figures. Okay, so this is the figure from your textbook. This is a figure that I always like in my microbiology courses as well. So basically, the first thing that a virus is going to have to do is it's going to have to recognize its target, and it will have to attach to that host cell that it wants to infect. And it's able to do that through cell surface receptors, okay? Now, once it's attached to that host cell, the next thing it's going to want to do is penetrate into that host cell. And that can include then an uncoating phase. So once it has merged with the outer membrane of that host cell and popped into that cell, it's going to lose its outer protein capsid or its protein shell. Okay, now once it's inside of that cell, I want you to think about, well, what does a virus want to do? Well, a virus, we said, is just nucleic acid and protein. So what's it going to want to do? It's going to want to replicate that nucleic acid, whatever it is, whether it's RNA or DNA. So it's going to want to replicate its genome and express it to also replicate and synthesize its protein shell. Now, once it has copies of its nucleic acid and its protein, what can it do? It can assemble new viruses, new virions, which are viral particles. Now, that's because you'll notice the virus does not have sex. It does not reproduce the way other cells do. It assembles new viruses. Now, once it has collected a nice aggregate or nice collection of new viruses, its final role is going to be to release those viral cells. And in order to release those viral particles, it will either do that by budding off, forming a little, envelope-covered virus particle. And that's how you end up with viruses that have envelopes, because viruses are jerks. When they destroy or when they're leaving a host cell, sometimes they steal part of that host cell's membrane. and it helps them get around undetected in the host body and infect other cells as well. Or it will simply completely lyse that whole cell and just burst out. Okay, and so this figure shows the same steps too, but I find this one a little bit easier to follow. Now, to give you a recap of those stages, I wrote out the recap here of the main steps. So again, recognize, attach, penetrate and uncoat, replicating the genome and making more proteins, assembling new viruses, and then releasing those new little viruses. Now, once a virus is infecting a host cell, there are two options for how that infection can go. One of them is the lytic infection, and when it comes to lytic means that a cell, in this case a host cell, is bursting, getting destroyed. And the reason that that happens is that the virus is replicating. Okay, so there's the assembly of a bunch of new viruses and it destroys the host when it wants to release those. Now, what's different in terms of lytic versus lysogenic? For lytic, I want you to remember the reason the host cell is bursting is because not only do you have the virus getting that host cell to replicate the viral genome, but it's also expressing that genome. Okay, so it's expressing the genes of the virus, making the proteins of the virus, and assembling new viruses. Okay, that's different because that's a nice active infection. That's different from lysogenic viral infections because lysogenic is basically dormant. You have the virus in those cells, but you don't know it because the only thing that that virus is doing in the lysogenic phase is having the host cell replicate its genome. Okay, so it's replicating its genes, but it's not doing anything with them yet. So you don't know the virus is there because when it's replicating the genome, there's no product yet. It's once you have protein products, gene expression and new viruses getting made, that you're then in the lytic phase and it bursts through the cell. Okay, so viruses will cycle through going back and forth between lytic and lysogenic. And please remember the difference is lytic, you have the proteins being produced and new viruses being assembled. Lysogenic is dormant, you only have genome getting produced. Now, when we talk about viruses, there are a ton of different viruses, huge variety. And as you can see with this table from your textbook, there's a lot of different ways that we can categorize and name viruses. So viruses can be categorized or separated based on whether or not they have an envelope around them. So the envelope versus the non-enveloped viruses. And you see that distinction here again. And when it comes to envelopes, like I mentioned earlier, a lot of times that can also be a stolen outer membrane from a previous host cell. And then the other way that we like to categorize viruses is based on what genome they have. So do they have DNA or do they have RNA? And sometimes you'll see them distinguished as whether or not they have single-stranded or double-stranded genomes. Okay, and the third option is sometimes they'll be categorized based on shape, okay, whether they're helical, icosahedral, but that you don't have to worry about for our purposes. Now, what's interesting about viral infections is that the consequences of the virus can vary depending on what cell type it's infecting. So the figure here is pretty cool because it shows the same virus, which is simian virus 40. It shows the difference between what this virus does as its life cycle when it's in a rodent cell versus when it's in a primate cell. And it's kind of like the difference between lytic and lysogenic and various consequences from that. So you'll notice that when simian virus 40 infects primate cells, because simian means monkey, what happens is a lot of times it will infect the cell and then it will basically have that host cell express the viral genes to make viral proteins. and then assemble new viruses. And as you saw in the previous slide, once you have the assembly of a whole bunch of new viruses in a host cell, boom, it fills up that cell, lyses it, explodes out. But now you see the difference in rodent cells is a lot of times in rodent cells, instead of killing the cells and being actively, assembling viruses, notice that instead the large T proteins gene that triggered the crazy explosion in the primate cells, at this stage in the rodent cell, instead of getting actively expressed and making new viruses, the large T protein gene instead gets integrated into the host cell DNA. And now the host cell replicates and replicates that viral genome. And what's interesting is that it basically triggers transformation. And in this case, it's not like the transformations that we do in the laboratory. This term transformation here means that it causes the cells to mimic cancer, to basically be cancerous cells, having uncontrolled cell division now. Okay, and so the large T gene, because it can trigger this transformation of rodent cells to make the cells cancerous and have uncontrolled cell division, it's known as an oncogene, okay? A gene that can cause cancer or tumor, usually by causing uncontrolled cell growth, okay? So in this example, the large T gene is the oncogene, and over here you end up with cancer in those cells. One of the types of viruses that you hear come up a lot are retroviruses. The major example of this, which I'll have on the recap slide coming up, is HIV. Now, with retroviruses, their genome within them is single RNA, and it's surrounded by two protein shells and an outer envelope. So lots of outer proteins around them. Now, the big problem for us is that retroviruses, because it has an RNA genome, they use reverse transcriptase, which is an enzyme that allows them, once in the host, to have that host make a DNA copy of their RNA genome. Now, that's very problematic for the host because reverse transcriptase, when it's making a DNA copy, it's really bad at its job. It makes mistakes and it has no proofreading ability, kind of like some of my students. Now, as you know, when you don't proofread, you don't catch the mistakes that you've made. Now, this is a problem when it's doing this with DNA in a host cell because mistakes in DNA are mutations, right? So the reverse transcriptase is the reason why retroviruses like HIV mutate so much and so quickly. That's the problem that it causes for us, is it creates a bunch of mutations when it's replicating its genome. And that means that we can't pinpoint, target, and destroy that retrovirus because every time we know the sequence and we want to target it, well, now it just changed again because of all those mutations that build up so quickly. The other thing that retroviruses have is long terminal repeats, which are sequences in its DNA copy that allow that DNA strand to integrate into host genomes, meaning the long terminal repeat sequence allows that piece of viral DNA to cut open your DNA sequence as the host and embed itself, glue itself into your DNA sequence. So then the viral DNA is now merged with your DNA, and you now make tons of copies of that viral sequence for the virus. The last point that I want to make is they have these genes that you see in the figure, CP, RT, and EP. And some of it we've been discussing by discussing the form of the retrovirus. CP stands for the coat protein, so the two layers of proteins around the genome, so yellow in this figure. Reverse transcriptase codes for that enzyme that allows for replicating the viral genome and making the DNA copy. And then EP codes for the envelope protein that makes the outermost shell or envelope around the retrovirus. Okay, so CP is coat protein, RT is reverse transcriptase, EP is envelope protein. And here's a recap of the notes that I just went over on the previous slide. Now, in order to really understand what viruses are doing to you and to understand cancer as well, it's important to have, a quick crash course in terms of your immune system and your immune response. So I'm just going to go over a few slides to help you visualize what your immune system is doing for you. When it comes to your immune system, there are two forms of immunity that you have. There's innate and there's adaptive. And like the term suggests, if something is innate, it means you're born with it. So innate immunity are kind of like the barriers, the very quick barriers to help protect you against microbes that you are born with and that help protect you from any type of microbe without your body specifically identifying and targeting that particular microbe. Okay, so for instance, mucus in your nose, like snot, you may think that it's gross, but it's actually part of your innate immune response so that any of the microbes that you're breathing in will hopefully get stuck in there, and then you sneeze them out or you blow your nose and you get them out of your body. Okay, so it's non-specific, it's not targeting a particular microbe. It never gets to know the name. It doesn't distinguish, oh, this is COVID, oh, this is a cold, the flu, this is, a bacteria. Okay, it just tries to block anything it can. Then you have your adaptive immunity. That is what you build up over time. Okay, it's much slower than your innate immune system, and it's highly specific. So it's slower because it takes the time to identify the exact microbe that you're dealing with, and it makes specific antibodies to help you fight that particular microbe, whether it's a specific virus, a specific bacteria, and then it creates what we call memory. So it saves some of those adaptive immune cells that it developed for that particular microbe. And this is why things like vaccines work, okay? Vaccines are working on your adaptive immune system, your body's ability to take a look at a particular cell, take a little piece of it, and make antibodies and make an immune response targeted for that exact microbe, okay? So that's the main difference between innate and adaptive immunity. Now this slide is just to help you kind of start to recognize some terminology that you might encounter in this course and especially in other courses that you take. It's an overview of all of the different immune cells or immune system cells that help protect you as the innate immune system, as part of the adaptive, and some of them as part of both innate and adaptive immunity. Now for our course, so I'm not going to have you memorize all of these or go through the crazy details of all of them, but I do want to right now have you circle star highlight NK, the natural killer cells. Okay, the reason why I want you to circle star highlight this one is in a few minutes we're going to start going over cancer, and you'll notice that NK cells can kill cancer or tumor cells. They can kill your infected cells and they can kill cancer tumor cells. So for NK, natural killers, I want you to circle star highlight the fact that they can kill cancer cells. Now, before we move on to discussing cancer, I just want to give you a brief summary of what exactly that adaptive immunity is. So please remember, we already mentioned that adaptive immunity is the type of immunity that changes and grows throughout your lifetime. It's basically adapting based on whatever microbes you have encountered at each step of your lifetime. It's basically why, you can't get badly sick from the same infection, twice throughout your life in some cases. And basically, it's the reason why vaccination also helps to protect you. Now, there are two parts of adaptive immunity that I want you to know. Whenever you hear humoral or cellular immunity, please think of adaptive immunity. These are the two halves of adaptive immunity. Humoral immunity is your B cell response and the production of antibodies, whereas cellular immunity is the T cell response. And we're going to go through exactly what this looks like. So here I mapped out for you what humoral actually means, how those B cells and antibodies get produced. And then the cellular side is focusing on your T cell response. Both of these together make up your adaptive immunity. So you'll notice that both parts of adaptive immunity start with the same type of cell. It's the helper T cell. And basically what happens is antigen presenting cells will show the helper T cells what microbe is currently invading your body. So for instance, if you have a COVID infection, it'll show, an antigen presenting cell in your body will grab a hold of one of those spike proteins from the COVID virus, and it will hold it up to your helper T cells so that they know that that's the bad guy to go after. Now, your humoral response, we said, focuses on B cells and antibody production. So once the helper T cell knows what microbe it has to target, it then stimulates the differentiation of B cells into what are called plasma cells. And those plasma cells are the type of cells that will then produce antibodies that are specific to the microbe's antigen. So for instance, if it's the COVID spike protein antigen that the T cell has been shown, then the antibodies produced by the plasma cells will target and be specific to binding that particular spike protein. At the same time, those helper T cells will also help activate macrophages and other immune cells. And what that means is those are immune cells that will help engulf some of the microbes that are currently invading. Now, at the same time, you have the cellular response going on, and that's basically when the helper T cells are promoting the production of cytokines. And cytokines are important little molecules or chemicals within your body because they trigger what's called the differentiation of cytotoxic T cells. And I want you to underline cyto and then circle toxic, because cyto, what this means is cell, and toxic, as you know, means something that's harmful or will kill. So the cytotoxic T cells will release toxins that trigger apoptosis. And apoptosis is important when you're infected because it will help in the destruction of viral infected cells. So your own host cells that have been taken over by viruses, your immune system will target for destruction and excretion from your body so that it can help put a stop on the virus factory that your cells have been turned into. Now, what's important about adaptive immunity, as I mentioned earlier, is that it has memory. So basically, when your body is cleaning up this adaptive response and getting rid of, the remains of those discarded apoptosis destroyed cells and getting rid of some of those T cells and B cells that have taken over, all the space and resources in your body, your adaptive immunity will save some of them. Okay, so over here, this is very important. Circle star, highlight this fact because it tells you why vaccines work and why you will not get as sick the second time you encounter some pathogens. It's because your body took the time already to make all of the necessary T cells, B cells, antibodies specific to that pathogen. And now you have some of them saved in your body. So it doesn't take, 4 to 7 days that it usually takes for your body to recognize and make the proper differentiation of B cells and T cells and all that. Instead, you have some saved up. If you breathe in or encounter that pathogen again, these can quickly target it rather than having to take those very tiresome initial steps. Okay, so now as promised, we're going to head into a discussion about cancer, what it means, and some of the implications or consequences of cancer within the body. Now, this is one of the slides that I will have a recap on the next slide, so don't worry if you can't jot everything down. When we talk about cancer, the first thing I want to mention is, well, what exactly is cancer? I know it's a term that unfortunately all of us have probably had to deal with in one way or another with either ourselves or a loved one. Cancer is one of the leading causes of death in the US and in the world. And ultimately, the way you can define cancer is the uncontrollable growth and division of cells. So you have uncontrollable cell growth and division leading to these getting, tumors getting larger and larger out of control. Now, there are 6 characteristics that any cancer cells will have in common. So whether you have someone dealing with brain cancer, breast cancer, lung cancer, pancreatic cancer, any of the different cancers that are unfortunately out there, they will all have these 6 general characteristics, okay? The first one is kind of like I was just saying, their cancer cells will have uncontrollable growth and division in situations where normally cells would be stopped. Their growth and division would be under control and they would have a pause signal. Now, related to that, the second aspect that cancer cells have is that even when their neighboring cells send out what's called stop growth signals, meaning, normally if cells are getting too crowded in together, neighboring cells will secrete signals that tell those cells to stop growing. There's not enough room, there's not enough resources. Well, cancer cells basically don't listen to that. They think their neighbors are crazy and they don't listen to them. So they keep growing and growing. The third thing that makes a cancer cell cancerous is that in addition to not listening to those other stop signals, they also avoid what's called the self-destruct signal that usually happens when DNA damage is detected in a cell. So they basically can override stop growth signals, they can override self-destruct signals, and they can just keep going and going. Interestingly is the fourth characteristics that they have, while they keep growing and dividing, they're able to hijack your vascular system, and they could actually cause you to grow new blood vessels so that now their larger and larger tumors can be supplied with nutrients. So they basically hijack your vascular system. And they are, fifth characteristic, they are basically immortal, okay? Because normally a cell can only divide a certain number of times and then it has to be destroyed, whereas the cancer cell can just keep growing and growing and dividing much further than a normal cell can. And then that brings us to the 6th characteristic, which is the most dangerous and the reason why so many cancer deaths end up occurring. The 6th characteristic is that cancer cells are capable of metastasis. And when you hear metastasis or metastatic, that means that they can travel. So if you have this cancer or primary tumor initially stuck to, let's say, your skin as a skin tumor, well, then they can have some of their cells break off, enter your bloodstream, end up going circulating to other places, and boom, suddenly that person has, let's say, a brain tumor or a lung tumor that comes up that initially started simply as skin cancer. And so when I say 9 out of 10 cancer deaths are due to what? It's due to the cancer having metastasized. So unfortunately, once it gets to that stage where it's become metastatic, it is usually too late to do anything for the patient. Now, as promised, here is a recap, including the definition of cancer, the 6 characteristics that cancer cells have, and then highlighted the aspect of metastasis and how that's the main cause of cancer deaths. So now when we talk about cancer and what's causing cancer, a lot of times, people will say, oh, this causes cancer, that causes cancer. For instance, like my mom is one of the people who doesn't want to use a microwave because she says it causes cancer. Now, when you hear those causes of cancer, like smoking, for instance, the fact is that cancer is ultimately a DNA disease. So yes, all of those things like smoking will contribute to the cancer, but the reason why they're contributing to that cancer is that they trigger mutagenesis. So cancer is ultimately a DNA disease, meaning that the true cause is that all of those bad things that you hear about can trigger mutations in the DNA of cells that then cause problems for these 2 types of genes, tumor suppressor genes and oncogenes, and then that leads to the uncontrolled, cell growth and uncontrolled cell division. Okay, so those 2 types of genes that we're going to go more into detail in the next slides, basically tumor suppressors, they're genes that make proteins that are supposed to, stop cells from dividing and stop cells from growing. And so if you have a mutation blocking those, now the cell will keep on growing, keep on dividing because it loses its stop protein. Oncogenes, the second type of genes, are the ones that I mentioned that basically are known to cause cancer and tumors. That's because they basically stimulate growth and cell division. Okay, so in general, tumor suppressor genes and oncogenes are opposites. They have opposite functions. But now if you mutate them, the tumor suppressor genes that are supposed to stop growth and division can now cause more growth and division. And oncogenes that already stimulate growth and division can be stimulated beyond control. So too much growth, too much division. And either way, you end up with cancer. So when we talk about oncogenes, before we get into proto-onco and onco, I just want to mention the Rheus sarcoma virus. And what the Rheus sarcoma virus was the first retrovirus shown to cause cancer. And now we've seen that a lot of viruses can actually cause cancer. We'll talk a little more about it in later slides, but basically a lot of their mechanisms for integrating into DNA and being able to jump around within DNA sequences can cause a lot of problems in your DNA. And as you know, problems or issues in your DNA end up being mutations. Now, when we talk about oncogenes, we keep saying these are genes associated with cancer and causing cancer. And 2 terms that you'll see are proto-oncogenes and oncogenes. And the difference is that the proto-oncogenes are basically the normal version of the gene. So proto-oncogenes are normal, they're necessary for the proper regular growth and proliferation of the cells. And then what happens is some sort of transforming event or mutation causes the proto-oncogene to go out of control and to become an oncogene. So for instance, it could be a virus like the Rheus sarcoma virus triggering that proto-oncogene to become an oncogene, which is then you can think of it as the messed up version that is, basically causing uncontrolled cell growth and proliferation. And I want you to circle, star, highlight, MAPK, as I call them. MAPK pathway and genes are mitogen activated protein kinase pathways, and a lot of the proto-oncogenes that become oncogenes are part of that pathway, because that pathway, which is shown in this figure and listed out in these genes over here, they are responsible for the cell functions that include cell growth, cell division, which is proliferation, and apoptosis. And so when you get those pathways, the genes in those pathways mutated and messed up, well, now you're going to have issues in proliferation and, programmed cell death, which means you'll end up in a cancer situation, because that is what cancer is. It's uncontrollable cell growth, uncontrollable cell proliferation, and a major lack of proper apoptosis or proper destruction of cells that should be getting destroyed. And instead, they're just growing, replicating, and taking over the body. Then we have the other types of genes that tend to be messed up in cancer, and they are the tumor suppressor genes. And I know when you hear tumor suppressor, you're like, wait, that's a good thing. That should be preventing cancer. And it does when it's working properly. But the problem in cancer is that these tumor suppressors get mutated, messed up, so they're not doing their job anymore. So when you think of tumor suppressors, they're actually good and important genes that produce proteins that help inhibit the replication and proliferation of cells. And one of the big ones is P53. So as you can see here, P53 circle star, highlight that guy there, because that one ends up being a huge implication in many, many different cancers. It's actually found to be in more than half of all human cancers. So when that gene is functioning the way it's supposed to, it acts as a tumor suppressor, and it blocks and prevents uncontrolled cell growth and division. But then when it becomes mutated, it ends up not being able to slow down cell division, and it ends up no longer promoting cell death the way that it's supposed to. Okay, and so that becomes very problematic. As you can see, that basically leads to the definition of cancer. The last thing that I want to talk about before we move on to the next topic is to now connect the two categories that we've been talking about, connecting viruses and cancer. And I'll have a recap slide in a minute to help you grab all of these notes. So the first thing we want to ask is, well, how exactly can a virus cause or lead to cancer? And there's a few different ways, some of which I've kind of mentioned already. First of all, some of these viruses, like the retroviruses, are, so ineffective in terms of the enzymes that they hijack or that they use, that leads to high levels of mutations, like the reverse transcriptase mutations of retroviruses. Another aspect is the fact that we said a lot of these viruses, especially having LTRs, they can cut open your DNA and insert their own sequences of DNA within your DNA. Now, when they're doing this, it's not like they have eyes and can say, oh, hey, that's critical for that host cell, or hey, that's a proto-oncogene or an oncogene area. Let's not, go near there. No, they just cutting into your DNA and jumping around. They can be transposable, meaning they can move from sequence to sequence. And when they're jumping around and embedding into your DNA like this, they can mess up some of your own genes. And if they're messing up your own genes, what does that mean? That's a mutation. So ultimately, those mutations can then lead to cancer. Now, when we talk about viruses leading to cancer, I want you to circle, star, highlight this second bullet point, which is asking you which virus is the leading cause of cervical, throat, and rectal cancer. And it is HPV, human papillomavirus. If you take microbiology or virology, we go into a lot more detail with that virus, especially since it is unfortunately killing a lot of people, especially people who find out that their partners, their monogamous, or so they thought monogamous partner was cheating on them, and now they have HPV and ultimately end up dying from cancer. Also, people, because viruses like HPV stay in your body long after you've actually been infected, you can be happily married to someone and not realize that back in the day they may have been promiscuous. They may have slept around with the wrong people. They picked up HPV back then, and then they give it to you once you are happily, safely married. So you could have been a virgin your whole life and then pick up HPV through marriage and then ultimately end up with these very dangerous cancers. Now, there are a lot of different ways that we can fight cancer, although unfortunately there are a lot of times when we do not win that fight. Some of the traditional approaches that you've probably heard of are surgeries, radiation, chemotherapy, and treatments with antibodies that specifically target the tumors. But as you probably know, a lot of these treatments can end up doing a lot of side damage to your body and can really beat up a person and leave them immunocompromised and in pretty, bad shape in our attempt to fight the cancer. Now, because we've mentioned P53 in the previous slides, that's another target that they're sometimes using for treatment now. A lot of research is focused on trying to find ways to reactivate or restore function of P53, since you saw that in more than half of the types of cancers that we know of, P53 tends to be involved, especially in loss of function of P53. So if we can restore that function, we can really help to fight cancer. The last type of treatment that's been emerging ever since the 1990s is called virotherapy, where they're actually using viruses to try and target the cancer. So in some cases, they modify the virus, especially its receptors, so that it's more likely to recognize the receptors on cancer cells and infect and burst those cells. They also basically try and modify viruses to be more active in cancer cells so that they're destroying the cancer cells rather than the human host cells. And here's a recap slide for you to pause and jot down notes. So now we're going to switch over away from viruses and cancers and switch to a completely different chapter or completely different topic, which is glycolysis. And the rest of the semester mainly focuses on energy creating pathways like glycolysis and electron transport chain and Krebs cycle and photosynthesis. So this is where we're kind of heading now for the rest of the semester. So as we head into the glycolysis section of this lesson, it's important to first say, well, what exactly is glycolysis? And whenever you've done chapters like an intro bio on cellular respiration, you'll notice that glycolysis was listed as the first phase of that. It was anaerobic because it didn't require oxygen. It was the only part that occurred outside of the mitochondria. So now when we look at glycolysis, what it actually is the breakdown of the 6-carbon glucose molecule into 2 molecules of pyruvate, each of which are 3-carbon molecules. And in the steps of glycolysis, there will be an important intermediate, which is fructose 1,6-bisphosphate, which we'll talk about a little bit later on. So this portion here is glycolysis, making those 2 pyruvate molecules, which then serves as a very important product because it can go into various other cycles or stages to make a lot of energy for a cell and to do a lot of different things. So here you have the 3 main possible fates or pathways for pyruvate to then go into. You have anaerobic glycolysis. You have aerobic oxidation, which will go through all of the other steps of cellular respiration, which is the citric acid cycle and the electron transport chain to make a whole lot of energy for the cell. Or you have anaerobic fermentation. Okay, so basically you can have anaerobic conditions without oxygen, and if oxygen is present, then you can have full-blown oxidation and a lot more ATP created. And we'll go through all of these different options throughout the rest of the lecture today and throughout the other lectures that we have left for this semester. Now, as an overview of glycolysis and the various things we're going to go over, I want you to know that there are 10 steps in total in glycolysis. And we basically tend to split the whole process in half. And so we look at one point at the first 5 steps, and then we take a look at the last 5 steps, creating the ultimate 10 steps to go from glucose to pyruvate. So now, like I mentioned in the previous slide, because glycolysis has so many steps to it, again, remember it's 10 steps, and each step has its own enzyme. A lot of times when studying it or kind of researching it, it makes it easier when we split the steps of glycolysis in half, and we focus on thinking of the first half as the conversion of that glucose, which we said is 6 carbons, to a 3-carbon intermediate glyceraldehyde 3-phosphate. And so that will be the first half of the steps. And then the second half of the steps is then converting that 3-carbon molecule to pyruvate, which is the ultimate goal of glycolysis, because like I said, pyruvate is then very powerful because it can go into any of the 3 fates that we mentioned already. And one of the big ones is the rest of cellular respiration, so the Krebs cycle and electron transport chain. Now, when we think of these processes, 2 enzymes are going to be very important. The very first enzyme of glycolysis and the very last enzyme of glycolysis. And we're going to go over in later slides why they're so important. Basically, they're big time regulators of the whole process. So I want you to remember these 2 names. The very first enzyme in glycolysis, so the first step uses this enzyme, is hexokinase, which you can see here in this lovely meme. So hexokinase is responsible for the first step of glycolysis, and then pyruvate kinase, or PK, is responsible for the last step. And circle star highlight both of those. Know that hexokinase is the first step enzyme and pyruvate kinase is used as the enzyme in the last step, because those are going to be important in a few slides. So now again, looking at glycolysis as the first half and the second half, here we have mapped out the whole first half, which means the first 5 reactions or first 5 steps in glycolysis. And those first 5 steps, this part of the glycolytic pathway is basically to prepare for the electron transfer and the eventual phosphorylation of ADP. It's kind of like a prep phase of glycolysis. And again, you'll note that the first enzyme is hexokinase to convert glucose into glucose 6-phosphate. And so that's an important one to remember, because that will be a regulator and will be inhibited in later slides that we talk about. You'll notice that for this first half of the reaction, it's mentioned over here that 2 molecules of ATP are required for these reactions to occur. So this is the part where you will be putting in energy to eventually make energy later on. Now, when you see any of the kinase enzymes mentioned, so you see it here and you see it here again, please remember that kinases are any of the enzymes that are ATP dependent and will transfer a phosphate group from ATP to whichever substrate that they're phosphorylating. So that's why you see that ATP is required here and ATP is required here, because both of these enzymes are kinases. You have hexokinase and you have phosphofructokinase. ATP is required in these steps because the enzymes will need to take a phosphate from that ATP and use it to add the phosphate group to the substrate to create that next molecule in the glycolytic pathway. Now, what's interesting is that kind of like we discussed when we talked about enzymes. A lot of enzymes work with negative feedback pathways. So the hexokinase enzyme that I keep mentioning is actually inhibited by glucose 6-phosphate, because once you have a lot of that product, you no longer need hexokinase to keep making more of it. It would be a waste of resources for the cell to keep making more of it, and a waste of energy, because as you can see, that reaction requires ATP. Then we have the second half of glycolysis, and what you'll notice in this second half is the ultimate goal is to create that pyruvate that we mentioned before. And you'll see that now, instead of using up a bunch of ATP, now you are getting the production of a lot of ATP throughout this process. So the second half is about making the energy, and the first half was about, using energy in order to prepare for this second half. And again, what I also want you to notice is that each step in these reactions has its own unique enzyme. And so that's important to note, because when you then want to control the steps or control a pathway like glycolysis, a lot of times it's about targeting the individual enzymes. And I want you to circle, star, highlight that pyruvate kinase is the very last of the enzymes in glycolysis, because just like hexokinase was the first one, basically an important control point to be inhibited, pyruvase kinase is another control point to be inhibited, and we're going to talk about that in a few minutes. The last part of the glycolysis pathway that I want to mention is called anaerobic metabolism of pyruvate. So back when we showed 3 possible fates of pyruvate, the first one was basically anaerobic glycolysis, the anaerobic metabolism of pyruvate. Now this reaction reduces pyruvate to the product called lactate or lactic acid. It's an exergonic, so that's this guy here, exergonic reaction, and I'm going to have a recap of this slide in a minute. And the enzyme that it uses to do this is called lactate dehydrogenase. So you'll see that as LDH in various places. Now, there are 2 clinical connections that I want to make regarding the metabolism of pyruvate, specifically the anaerobic metabolism that produces lactate or lactic acid. So the first connection is to dentistry. And if you're someone who eats a whole lot of sugar, that's going to be problematic for you, because, bacteria, they love sugar, and that sugar content on your teeth will promote the growth of bacteria into what's called plaque. And plaque is basically an aggregate, a big clump of all different species of bacteria working together to create this massive colony of growth on your mouth. Now, if you picture that, and I know you don't really want to picture it, but if you picture a big clump of all these bacteria, well, the ones all the way underneath the surface of this big sticky clump, they're not going to have access to oxygen. So they're going to use this anaerobic metabolism of pyruvate. Now, we said, what's a product of this metabolism? Lactate, which is lactic acid. And when you hear the term acid, that tells you that this strong organic acid is going to break down or destroy the enamel on your teeth. And you could end up with a lot of problems, including the painful cavities that you can get. Another clinical example that I want you to be aware of is how anaerobic metabolism relates to cancer. And what you're going to see is this figure here demonstrates what's called the Warburg effect. And whenever you hear the Warburg effect, I want you to note that that is basically how cancer cells, which is the one on the right, even when oxygen is present, have a preference for anaerobic metabolism, where they go into this increased rate really speedy rate of glucose uptake and production of lactate through anaerobic metabolism. Okay, so they'll do that instead of going the aerobic cellular respiration route that the normal cells would do. And people, finding this out about cancer cells has actually helped in terms of diagnosing and even looking for treatments in terms of cancer, because first of all, for diagnosis, they can look for this crazy Warburg effect. They can basically look to see if you radioactively label an analog to glucose and track it in the cells, you can see if there's this massive quick uptake and anaerobic metabolism with a high production of lactate as a byproduct. And that would tell you that cancer is present. It can also be useful in terms of treatment because now they can look for, inhibitors or activators involved in this Warburg effect, involved in this particular pathway to try and knock it back to the normal, proper pathway. Okay, so like I promised, here is a recap slide. Some of the key points that I just mentioned on that previous slide. Now, when you're talking about any kind of reactions in chemistry or biochem, you have to ask yourself, is this reaction endergonic or is it exergonic? So endergonic would mean that you would see a positive free energy, and it would basically be a reaction that's absorbing and requires energy, whereas exergonic would be a negative value of the delta G free energy, which you could look through these values to see any of the negative ones would be exergonic. You have that decrease of free energy, which is basically like saying there's a release of energy. And when we look at glycolysis to kind of analyze whether it's endergonic or exergonic, it's interesting because glycolysis isn't one reaction, it's 10 different steps. And so some of the steps you'll notice end up having negative free energy values, whereas other ones end up having positive free energy values. So that shows you that some of the steps in glycolysis are exergonic and some are endergonic. And I like this figure because it basically shows you the reason that you have both present in glycolysis is you have to use or invest a little bit of ATP in order to make some ATP. And so when we look at all of these steps, you can then ask yourself, well, which one is it more? So is there a greater amount of the negative or a greater amount of positive? And if you look through all of those different free energy amounts and you add them all together, you'll see the overall values are actually very negative. So that tells you that overall glycolysis is exergonic. So it includes steps that are both endergonic and exergonic, but circle star highlight the fact that overall glycolysis is exergonic with a large decrease in free energy, and that indicates that it is a spontaneous reaction. So any of the exergonic reactions, exergonic steps, they happen spontaneously. Now, when we are looking at that, ultimately note that some of these steps are driving some of the other steps, and you'll see that it's mainly about 3 of the steps have much greater negatives than the others. So like steps 1, step 3, they're very highly negative, and also step 10. So these are very, very powerful driving forces for the glycolysis process. Now, like anything in life, too much of a good thing can become a bad thing. And in biology, we always have to have control. We have to be able to control and regulate any kind of pathway. And so the same is true with glycolysis. You don't want a cell to be just, undergoing glycolysis nonstop, that would be a big time waste of resources and energy, because we saw, not only does it make some energy, but it does take some energy and a whole lot of resources and enzymes to actually complete glycolysis. So when we look at the control of glycolysis, I want you to think about, some of the steps, some of the key enzymes that we've mentioned already. And when we talk about controlling a pathway, there are usually three best places to target. Okay, and I'll have a recap slide in a minute. The three best places to target any pathway is at the very start, the very end, or the intermediate. Okay, and so in the case of glycolysis, we said the very start, the first step to look at is right up here. So one of the first areas that we would block in the case of glycolysis is the first step, which we said, what enzyme does that? Hexokinase. So if you inhibit hexokinase, which we said earlier is actually inhibited by its own product, glucose 6-phosphate, you've now saved yourself from wasting all of the resources and energy of the next 9 steps that come after that first step. So blocking the first step, specifically hexokinase, is actually one of the best, most efficient control points. Now, sometimes instead, what will be blocked is the intermediate, which is this step here. So the production of fructose 1,6-bisphosphate. And that, as you can see by the figure, is blocked by ATP, which you're going to see in a later step. ATP also blocks the last of the enzymes as well. So sometimes there's more than one thing that can block these enzymes, but this figure points out ATP for each of those. And so with that last step, like I said before, sometimes instead of stopping something at the very beginning, instead you stop it at the very end, because that's the point where you realize, well, the whole point of this was to make some ATP and make pyruvate. And once you have pyruvate present, that sometimes is a signal to say, hey, okay, we got our final product, so let's stop right before then. And so this here, what is blocked here, is the PK enzyme, the pyruvate kinase that I told you earlier to please make sure you remember. That hexokinase is the first enzyme, pyruvate kinase is the last one. And the reason why I kept emphasizing them is those are two very big control points to turn off glycolysis and save the cell some energy once it's produced, the right amount of ATP and pyruvate that it needs at that moment. Okay, and so that's a very, very important step of control. Now, I want to point out, in addition to stopping or controlling glycolysis at the level of the various enzymes and through the presence of their particular products, you can also control glycolysis by the amount of, the starting reactant that you need. So by the amount of glucose, specifically the blood glucose level. And so you'll see that in addition to these enzymes and, the products being ways to control glycolysis, there's also a higher level of control by your natural hormones, specifically things like insulin that regulate the level of glucose in the bloodstream, which will then either activate or deactivate the metabolic pathways that are needed at that moment to handle the glucose. And that's what this little figure here is basically showing, how glucose levels can affect whether or not the pyruvate kinase is activated or deactivated based on what you need at that moment. If your blood glucose is low, or if it's high and you want to break some of it down. Okay, so a lot of that aspect that you see here is basically control of the liver pyruvate kinase by phosphorylation and by dephosphorylation. So as you can see, one is less active than the other form of that enzyme. And so that will be affected by the amount of glucose present in the blood. Now, as promised, here is a recap because that was quite a bit of information. Here are the key points that I want you to have in your notes for the control of glycolysis. And that is it for today. As always, if you have any questions or concerns, please contact me in either the Remind app or through email 24/7. Like I always say, I have no life and I am happy to help you. Okay? Thank you and have a great day.